We're very happy to have David Brady, who gave a wonderful introduction to the awardee, Dr. Diadamo, yesterday. As you know, Dr. Brady has worked with Dr. Diadamo for decades, and I was really sorry to hear. If I had heard about it, I must have forgotten that the Bridgeport College of Naturopathic Medicine actually closed. It was an unbelievable facility, but David's still a director at the Institute of Clinical Nutrition there, which is the premier one in the nation, as well as being a director, product development director at Designs for Health, and also the director at DSL, Diagnostic Solutions Lab, which is a great lab. So anyway, he's going to give us a lecture that's very enlightening as always. We really look forward to hearing him speak. And so without further ado, David, you're on.
Hello, everybody. Welcome back from lunch. Thanks, Woody, for having me here again. It's always nice to be back at IHS. Nice to be back in New York City. Nice to be back to normality, right? At a conference, seeing each other. And what I'm going to try to talk about today is something we see in our office all the time, particularly after the pandemic, even more so, which is mood disorders, depression, anxiety, stress, hypervigilance, PTSD. Really wraps in a lot of different things. And what we're going to try to do is use a multi-omics approach to try to help assess why someone may be having mood disorders, anxiety, hypervigilance, insomnia, panic attack, and then how to use the multi-omics platforms that are available to try to really personalize their treatment and come up with what would be the most effective approach for a specific individual. Because we know in those kind of disorders, not everybody responds the same to the same kind of intervention, right? Sometimes you got to do different things in different people to get the same positive outcomes, hopefully. We're going to touch on microbiomics, we're going to touch on metabolomics, and we're going to touch also on genomics at the end. And I only have an hour, so I can't make you an expert in any one of those domains if you haven't been using them already. But no matter where you're starting with some of these omics types of platforms, I hope to at least give you an idea how valuable they can be and to give you motivation to kind of go down that rabbit hole a little bit more and keep exploring those. My disclosures, as Dr. Merrill said, I'm the Chief Medical Officer of Diagnostic Solutions Lab in the diagnostic laboratory space, and for 20 years, the Chief Medical Officer of Designs for Health in the nutraceutical space. So the whole gut-brain connection, that seemed to be the buzz phrase, wasn't it, for the last few years, right? The gut-brain connection, gut-brain connection. What does that really mean to us, functionally, right, when we're trying to treat patients? Well, I'm going to try to show you at least one application of gut-brain connection, and that is how it is involved in mood, behavior, and aberrations of brain function, which may manifest in different ways: anxiety, panic, insomnia, depression, etc. And although, we know about the vagus nerve, that's another big kind of buzz topic right now in vagus nerve therapy and vagus nerve stimulation, which all can be very, very beneficial in these type of conditions. And it is sort of the direct connection or pathway or physically tangible connection between the gut and the brain. But it's certainly not the only connection between the gut and the brain. There's a lot of complex things going on. You have changes we're going to look specifically at changes in the microbiota composition in the gut and how that can affect brain function and brain chemistry even. So we're going to look at how microbes can change cytokine expression in the enteric immune system, which can affect the brain and potentially inflame the brain, cause microglial inflammation and functional changes in how the brain processes. We're going to look at how microbes may produce different neurochemicals, neurotransmitters, if you will, and hormones, or how they may consume them, how they may produce substrates for neurotransmitters or consume substrates for neurotransmitters, all potentially altering functionality. And it's a bidirectional system, I should say. Changes in the gut environment, such as the microbial composition in the gut, can have actual changes in how the brain performs, but changes in the brain through things like emotional stress and other types of just mental states of the person can actually also then alter the microbiota composition and how the gut's functioning. And, we're just really learning the scientific nuances and the minutiae of these connections, but we've inherently known about these connections for a long, long time, right? I mean, everyone has known for generations, many, many generations, that if you're stressed out, it can cause problems in your gut, right, and cause an ulcer. Like the gut is sort of the reservoir for stress that's going on in higher centers. And, in a person that's sort of well-balanced and generally in good health, and they have enough resiliency, they have a well-functioning HPA axis, they have normal behavior, they have normal nociception, they don't have exaggerated pain responses or global pain, they have a relatively balanced microbiota in the gut, they have normal gut function, they have a balanced immune system, and they're in homeostasis and they're pretty well. People who are under a lot of stress for particularly prolonged period of time or even severe acute stress of relatively short duration can rapidly develop a dysfunctional HPA axis, altered behavior, altered pain perception. They can go down that slippery slope toward global pain syndromes and things like fibromyalgia. They can develop things like IBS, panic attack, insomnia, and they generally have an associated dysbiosis. They have an imbalance in the gut that can actually alter their catecholamines, their neurotransmitters. They have functional gut problems, right? A multitude of symptoms that would be usually classified as IBS. They have low-grade or overt inflammation, tissue damage, and ultimately overt disease. Now, when we start looking at the GI microbiota and psychology, let's say, or psychiatry, these are not fields that generally intersect, right? The psychiatrist, psychologist is not usually concerned with what bugs are in your gut, and they're generally not taking stool samples, okay? But these things have touched in the past. For instance, the syphilis-causing microbe Treponema pallidum was responsible for filling large parts of Victorian mental asylums. So then there was a direct connection with microbiology and psychiatry. Psychiatrist Henry Cotton, he was the medical director of New Jersey State Hospital at Trenton, was convinced that bacteria in the teeth and in the oral cavity were the source of his patient's psychiatric conditions. One of the earliest kind of manifestations of the whole paradigm that oral health problems can create systemic problems. The thing we think of most now is, bad oral health, bad oral microbiota, and cardiovascular events and systemic inflammation, but it goes way beyond that, and it even can touch mental health conditions. And then probably the most modern direct forced connection here between microbiology and psychiatry was the neurocognitive effects of HIV infection. Became very evident, and again drew psychiatry into having to pay attention to microbiology. But it's all really just modern manifestations of Metchnikoff's philosophy, right? That if you have a bad gut environment, you just simply can't have a healthy person. Now we're just figuring out the little specific nuances and the details because of the Human Microbiome Project, and more specifically, looking at studies that actually typify what the microbial composition, or the GI microbial signature as they call it, is in depressed patients versus normal patients, or patients with anxiety versus patients without anxiety, and the like. So we're going to look at some of those relationships and how, knowing those things, we can potentially leverage that into different approaches for these disorders. Now, this is an interesting paper, Gut Microbiome-Wide Association Study of Depressive Symptoms. So in studies like this, and there are a multitude, I just presented this one here for you because it's rather new and it's a very, very well done study. What they were trying to do is what they often do in these types of studies. They're trying to use broad-based sequencing of the GI microbiota and just statistically come up with what's different about people diagnosed with depression as far as their microbial composition in the gut goes versus people without depression, right? And they do this for rheumatoid arthritis, and they do this for all these other diseases, okay? And it's useful from a metadata analysis research standpoint. It's not all that useful when it comes down to managing an individual patient, because what they're using here are sequencing techniques that don't have the fidelity to be able to get to the level of diagnosis and medical management decisions where you're saying, you know, Mary Jones has X microbe and we got to go get it, right? For that, you need more quantitative diagnostic methods like qPCR, but these are perfect methods to do these broad-based analysis to find out what's different in the guts of people with a certain disease versus without. And here they're really identified 13 different microbial taxa that were significantly and consistently different between people with depression versus people without. And you can look at some of the long names of those microbes. But what I'm going to really encourage you to do is to not get hung up on the names of the microbes and not get hung up on individual genus or species of microbes that may be different or not. It's more of the take-home message is at 30,000 feet. It's larger than that. Suffice it to say that we know that the composition of the microbiota in patients with depression is different than patients without depression. Don't get hung up on the individual organisms that are different, because these are not things that you can put in a pill and take generally anyway. They're anaerobic, and you just can't do it that way, right? But we do know that there's imbalance in the gut, and it's not in a favorable way. So that's the take-home message on that. And there's direct links that are shown. I'm going to lean on this study a lot. This is in the Harvard Review of Psychiatry. It's the title is interesting, Gut It, Unraveling the Role of the Microbiome in Major Depressive Disorder. And what I like about this study is it's a literature review. Basically, if you read this and look at all the studies presented and analyzed in this review, it'll save you from having to go read about 30 studies. And they did a very good job of pulling them apart and highlighting certain things. Like here, there's a direct link between the function of the HPA axis and how your stress functionally is processed in the body and your microbiota. And some of these are animal studies, admittedly, including this here, where they actually gave germ-free mice specific organisms, like in this case Citrobacter rodentium and Campylobacter jejuni, and actually then started observing behaviors in the animals that were indicative of depression and or anxiety. So they were able to actually induce these disorders by putting certain types of bacteria in the GI microbiota, which was pretty interesting. Now, this slide here I put in because I saw it a couple of weeks ago on Facebook as like a Facebook meme. And I think it was a promo for a lecture or something that somebody was doing about, you know, the importance of some of these organisms in different mood states. And it says here, your mental health begins with your gut. How? Lactobacillus makes serotonin. Bacillus makes dopamine. Bifidobacterium makes GABA. The health of your microbiome influences your mental health. And I don't disagree with the general take-home message of this. My disagreement with this is it's too simple. It's more complicated than this. It's not, oh, lactobacillus makes this, it makes serotonin. Give more lactobacillus and their depression goes away. It doesn't work like that, right? We wish it was that easy. It's not. It's helpful to get the idea across. It's just a little too simplified. In reality, this is much more complex. In healthy people, like we said, they generally have an intact HPA axis that's working. Their neural circuits are intact. Their immune system is generally balanced. Their microbiota is fairly balanced. In people who are depressed, they do not have a resilient, well-functioning HPA axis. There's disruption in their neural circuits. Their immune system is imbalanced, and it's imbalanced toward an inflammatory stance, and their microbiota is shifted and not normal. So we'll take a look at how they're shifted in some ways. I want to go back one. An interesting part of this slide, though, is in the bottom center there, where it talks about things that can actually alter the microbiome that may start, you know, maybe the dominoes where they start falling. Lifestyle, drugs, stress, diet, all these things are causal things to create shifts in the microbiota, but you can look at it the other way. They're also levers for us therapeutically to straighten things out and get them sent in the right way. So in this review article, Gutted, in the Harvard Psychiatry Review, they have a lot of these tables. These are taken directly from that paper. And this is looking at what exactly, through various studies, is altered about the microbiota in depressed individuals. And I just picked two here coming up. They go on and on and on in all these tables. And again, they're going to, through sequencing, tell you statistically which organisms are overexpressed, which ones are not. Which ones are there more than in normal people, which ones are there less. But the bigger patterns are probably more important. And generally what you find is reduced richness, reduced diversity, and a leaning toward inflammatory type of organisms with upregulation of inflammatory cytokines, like in the lower one there, increased levels of IL-6, IL-8, TNF-alpha, CRP, and then shifts, and we'll look at this when we look at metabolomics, shifts in the kynurenine-tryptophan ratio. So that involves the quinolinate and kynurenate pathway, which is neuroexcitatory, which we'll look at. But I put these two here because if you look at the upper one, it says no significant differences in TNF-alpha, IL-1B, IL-6 levels, and the diversity was significantly higher in this study. So I put it there just to illustrate that everything is not perfect and linear. There's conflicting information in the medical literature. So, of course, there usually is. So you have to take the preponderance of the evidence and the preponderance of the findings. And the preponderance of the findings and evidence, when you look at a lot of papers, is what it shows on the bottom. Okay, that there are consistent shifts. And not only do they establish that there's shifts in the microbiota, but if you give probiotics, right, commensal probiotics, what we think of as beneficial organisms that we would call probiotics, such as different species and strains of lactobacillus and bifidobacteria, it generally improves things. It improves the GI metabolome, it improves cytokine expression, makes them less inflammatory, but what's more important is in most of the studies it shows that their depression levels go down, that they don't have as much depression or they don't have as much anxiety and they're more functionally stable. Now sometimes these are studies directly looking at depressed populations, so it's a study looking at depression specifically, but in many of the cases where they've established these relationships, such as using probiotics and improving mood or depression or anxiety, were not studies on depression, they were studies on things like irritable bowel syndrome, or studies on chronic fatigue syndrome, or other chronic diseases that have depression and anxiety as a component of the symptom profile. And they teased out of it the fact that the intervention, let's say in this case, probiotics, improve depression. Now, if you look at these studies, you'll see, very specific genus, species, strain, right? But again, I'm going to encourage you to not get hung up on one magic probiotic genus, species, and strain and say in depression patients, I need to give them that, right? We know by nature research is reductionist, right? It's easy to keep it clean that way, right? It's also easier to get it through IRB that way, and there's a lot of other reasons why it is. But that's not how it really rolls out in clinical practice, right? I'm really more of Stig Bengmark's approach in probiotics, which is I'd rather build a beautiful symphony than have a great solo violinist, right? You're going to go further with the symphony effect than trying to find the magic star probiotic that's going to make all depression better. Okay, so the big take-home message here is that probiotics are helpful in these situations. So are prebiotics. Here's an IBS study using short-chain fructooligosaccharides showing improvement in anxiety specifically. This is one on resistant dextrin, improving depression, anxiety, and stress. So probiotics and prebiotics, there's significant literature. I'm just showing a little smattering of what's out there. If you do literature searches, can be helpful. But the heaviest therapy, the highest degree, hardest lever we can pull on actually changing compositional signatures of the microbiome is fecal microbial transplantation. And are there studies on FMT and depression? There are. And this is one here, one of the more known ones. This is with a single FMT procedure, and this was on irritable bowel syndrome, again, on men and women, and it improved depression and anxiety. So very interesting stuff. But to me, this is even more interesting. They're looking at a study here where they admit the depressed people have a different and compositionally distinct microbiome than people without depression. But that doesn't prove any causality. That simply proves an association, right? So they go further here, and what they actually do in this study, it's an animal study. It's an animal and human study. It's interesting. They took fecal microbial transplantation material from depressed humans and implanted it into animals and made the animals depressed and anxious, and they exhibited what they deem as increased anhedonia-like presentations, which means the animals were no longer capable of feeling pleasure, right? They couldn't have good feelings anymore. That's pretty cool. You actually take normal animals that they can functionally assess and objectively determine, how they're behaving, and they take fecal microbial transplant material from depressed humans, put it in the animals, and the animals become depressed. That's at causality right there, okay? But the general rule when you really tease all these studies out and you boil it all down is people who are depressed have a drop in alpha diversity. They don't have as many diverse microbes in the GI tract. And they have a increased relative abundance of microbes that are associated with a pro-inflammatory state and heightened inflammatory cytokine patterns associated with inflammation, which is not surprising, right? Because we know in the end, depression is not a genetic serotonin deficiency or a genetic Prozac deficiency, right? Depression at its core is an inflammatory brain condition, right? And in fact, many of the drugs that we think of as antidepressants, such as SSRIs, MAOA inhibitors, and things like that, we've always thought of them in their mechanism of action on how they manipulate serotonin or serotonin reuptake. In reality, many of these agents have direct anti-inflammatory effects, particularly on deep tissue microinflammation, like microglial inflammation, and many of them are actually antimicrobial. So that may very well be more responsible for how they act in depression than the mechanisms of action that we were taught in medical school. If you really want to kind of wrap your brain around the types of genus and species of organisms that in these studies showed positive changes in mood versus negative changes in mood, it really correlates with which are inflammatory and which are anti-inflammatory in their general nature and where they push the enteric immune system. And so those are listed there for you if you want that. Suffice it to say, as we did, that we're looking at microbes that are generally pro-inflammatory when we look at the delta or the changes that are going on in the microbiota. So Staph aureus, Candida albicans, B. fragilis, and so forth. But the microbiota doesn't just sort of generally drive inflammation or not. The GI microbiota composition has a lot to do with our core immune function and even our response to systemic viral infection. This is a really cool study, microbiota regulation of viral infections through interferon signaling. The gut microbiota modulates antiviral type 1 and type 3 interferon responses, ultimately regulating viral infection outcomes. The gut microbiota is capable of modulating interferon antiviral responses both in the gastrointestinal tract and extraintestinal sites. So it's sort of a regulator of our entire response to a viral infection. It becomes less surprising now that one of the predictive factors on how you ended up when you got COVID-19, whether you went down a critical illness path to, early ventilation and potentially death, or you were lightly symptomatic from that same organism as the other person has a devastating outcome from, was differences in your microbial composition in the gut. So when we start looking at these things from an omics standpoint, such as on the left here, quantitative PCR analysis, this is just one page of a report, but this is the page that has to do with common opportunistic organism overgrowth, including organisms that are specifically associated with inflammation and or with autoimmunity. You see here things like staph and strep, which are common organisms that can overgrow, but also things like Klebsiella, for instance, which we know is highly associated with autoimmunity. Klebsiella pneumoniae, specifically with rheumatoid arthritis, quite frequently. But in parallel, we can look at cytokine analysis. So this is a CytoDX cytokine analysis showing on the same person who has a microbiome which is dysbiotic toward an inflammatory pattern. Their cytokine analysis is also showing they're in a strongly pro-inflammatory state. So they do correlate very, very well. And one of the things we can do that's easy to kind of lower the inflammation that might be going on because of an inflammatory dysbiotic state is simply probiotics. Like we showed in those studies, it helps depression. It's helping depression probably most, there's multiple reasons how it could, but probably most by dampening this inflammatory response. So when you use probiotics, the simplistic way we probably learned it was that you're putting more ground troops in, right? You're filling up that gut with good bacteria, right? It's not really what's going on very much. The composition of your commensal organisms, meaning the good guys, right? The kind of things you're dealing with probiotics, is set very early in life. By the time you're about 2 or 3 years old, your commensal microbial signature is pretty much set. And it stays there unless you really do something actively to push it out of that envelope. Like taking tons of exogenous probiotics or radically changing your diet. Let's say you go totally vegetarian all of a sudden. That will shift your microbiota, including your commensals, at least to a significant enough degree to see on testing. But generally, you're not really changing that much. You can change a lot with the opportunists and even pathogens if you have an overt infection. It's really hard to change your core composition of your commensal organisms very much. And certainly when you do it, it's not sticky. In other words, you can give a lot of probiotic, like a multi-strain, multi-genus species strain probiotic at high dosages, and you can see the shift on lactobacillus, let's say, in a quantitative PCR test. But it's not universally changing all of the balance of the commensal organisms. And if you stop taking that probiotic, it will not be long before it goes right back where it was to begin with. It just doesn't have stickiness to it, okay? So it's not a numbers game, and it never was a numbers game, because when you look, even, I don't care how many billion CFUs the bottle impresses you by with the number, it's still the equivalent of taking an eyedropper full of green food dye and dropping it in the Pacific Ocean and expecting to fundamentally change the color of the Pacific Ocean. It's statistically insignificant. It's not going to do that. But you say, but they work. They do work. But they don't work on a quantitative, numerical, send in the ground troops basis. They're signaling organisms, right? They modulate how the enteric immune system behaves. Again, I'll bring up Sig Bengmark, one of the giants of probiotic therapy. You know what he calls probiotics? Pixie dust. Just a little bit. Massive effect, like butterfly effect, right? So it's not numbers. So you don't need to be so obsessed with the number of billion CFUs on the bottle. Because oftentimes the 100 versus the 50 versus the 200 doesn't really make that much of a difference, as long as you're sending the signal to calm down the cytokines. And that's what they're talking about in this paper here. So when we look at the microbiota also, we're looking at what's going on with these commensal and keystone organisms. You see in this patient right here, they have low commensals, right, including things like lactobacillus and Faecalibacterium prausnitzii, or FPRO, right? That's not a good state of affairs. That's generally going to probably be accompanied by an overgrowth of opportunistic organisms that are associated with inflammation, because nature abhors a vacuum, right? So something goes low, something else goes high, right, to keep it all in balance. But a couple of key commensals, Akkermansia mucinophilia, of course, has a role to play, particularly with modulating the mucus layer on the gut lining for good barrier function. Beneficial Clostridia classes by producing short-chain fatty acids. Faecalibacterium prausnitzii, really, really important and strong butyrate producer. Most of these commensals, though, again, you cannot put in a pill and give orally because they're anaerobic and they would die in the pill, right? It just wouldn't work that way. Akkermansia mucinophilia now is an exception. There's some technology out there that is stabilizing Akkermansia to the point where it can be delivered orally. They're working on FPROW as well right now, but it's just not universally available at this point. But you can still modulate these organisms by basically changing the substrate that is available in the gut for these organisms to feed on, right? You can do it with prebiotics. You can do it with resistant starches and things like that. But in the end, what boils out through this microbiome research is that FPRO is shown to be low in a lot of chronic illnesses, and depression is not an example. Here's citation on a study showing FPRO, or Faecalibacterium, being low in depression. And when we get up higher at about the 30,000 feet, level looking down, and you look at the phyla level or the family level in the microbiota, the two major phyla in the human GI tract are Bacteroidetes and Firmicutes, right? And we look at the ratio between those two. And they're like the Hatfields and the McCoys of the gut. Or I guess in more modern parlance, the Crips and the Bloods of the gut, right? You want to make sure both gangs have enough members to keep the other gang honest, and one is not overrunning the other, right? So you need enough members and within balance to one another to keep everything happy and in a homeostasis. In depression, this shifts, and it shifts to having more of the inflammatory Bacteroidetes and less of the anti-inflammatory Firmicutes. And you can see this on laboratory testing. Here's an example: high Bacteroidetes, low Firmicutes, and low FPROW, by the way, right above it. This is a classic type of inflammatory dysbiosis pattern that's consistent with a lot of inflammatory chronic disorders, but depression is certainly one of them. Here's another example just showing a low Faecalibacterium prausnitzii on another test. So interventions here include probiotics, and as I said before, I'm really more of a fan of a symphony. So this is an example of a probiotic that is 10, I think it's 100 billion CFU, I don't remember, but it's 10 strains, right, or 10 specific organisms. And when this was put together, it's put together intelligently by looking at each of the genus, species, and strains that are in it and really grading them on a sort of a report card of their ability to survive a low pH, or their pH resistance, they call it, their bile acid tolerance, their mucosal adherence capability, their lactic acid production status, and their immune modulatory effects, what they do to cytokine expression. And you just can't get all of the different functionality that you would want in one organism, right? That's why you put together these sort of groupings of organisms to get a global functionality of the probiotic product that is greater than any one organism can deliver. No matter how good there might be some research on that one specific strain in, let's say, IBS. It's good to use. I just wouldn't use it by itself. This is not a new concept. Over 100 years ago, George Porter Phillips put forward the concept of treating, called melancholia, right? Depression, basically, with lactobacillus. So again, this goes back a long way. We're just understanding the science behind it now. You can also use combinations of probiotics and prebiotics, right? Something like xylo-oligosaccharides, or there are many different types of prebiotics. As we know, If you're using prebiotics in patients that inherently have dysbiosis, including inflammatory pattern of dysbiosis, what's the problem with using prebiotics? It makes them bloated and it's not comfortable, right? So you can try to find one that works for them. Xylo-oligosaccharides is probably the least bloat-producing prebiotic that I've used clinically, along with Saccharomyces boulardii, Cerevisiae boulardii, right? Saccharomyces, as we know, is great to use to treat antibiotic-related diarrhea, to treat C. diff, and there's multiple mechanisms of action by which it works, but it's also just a really good prebiotic, actually. It's classified as a prebiotic as well because it promotes the growth of the good bacteria and tends to attenuate the growth of the bad guys. Diet's a really important lever. So you need to make sure the person, the subject who's depressed is eating a decent diet, right? And you got to pay also attention to the dietary folate and B12, or they need to be supplemented. And if they're seeing someone like us, they're probably going to be supplemented. But if not, they need to make sure they're having a diet that's getting that, because we know the importance of those nutrients in methylation and neurotransmitter balance. And the studies that have been done on different types of dietary interventions in depression do lean toward Mediterranean diet high in fiber and fish and all of those good things in the Mediterranean diet. Now, you know, we all love our pills and potions and capsules and all that stuff, but really the best place to get this fundamentally is through food, right? So the return of probiotic-rich foods, prebiotic-rich foods that are more traditional foods that kind of went away for a long time, and now they're coming back with a lot of people, particularly people interested in wellness, that's a good thing. Oftentimes you have to supplement still, but it can lower the burden of doing that. Resistant starches, I mentioned. You can get these, of course, in certain foods, a lot of different beans, legumes, bananas, and so forth. And then there's commercial resistant starch products on the market, which can serve as really nice prebiotic therapy to push the microbiome in the right way. And polyphenols are very, very useful. This is a polyphenol blend, for instance, with blueberry, cranberry, pomegranate, quercetin, broccoli, red grape powder. This is just an example of something, a polyphenol blend, that can help the microbiota push toward a healthy state. But if you have dysbiosis and you test it and you have a lot of opportunistic overgrowth, you have low commensals, you might have to go get the bad guys. You might have to shake it up, right, and try to restore better balance. You have a couple options in that. You know, you can use more natural therapeutic agents, or you can use pharmaceutical agents. You can use locally acting antibiotics like Xifaxan, rifaximin, or so forth, and they work. They get rid of a lot of stuff. In my experience with them, not only personally using them with patients, but talking to colleagues and looking in the literature, the Xifaxan, rifaximin kind of route, whether you're using it in SIBO or what have you, it'll knock a lot of stuff down, and often people feel better for about a month. And then it comes back, right? Because I think it's too indiscriminate. It's just taking everything out, and then how do you set up good balance just because you first wiped out a bad balance, right? It doesn't necessarily equate. I think the functional approach using botanicals, volatile oils, things like that, I think works better. And Jerry Mullen did studies at Johns Hopkins showing that they work pretty much equivalently in the short term, but the natural therapeutics work better in the long term because you don't get that refraction a month later and then they're back where they started. So using antimicrobials that are common in the whole functional medicine approach, whether it's things like Artemisia annua, berberine, berberine-containing herbs of various types, citrus seed extract, caprylic acid, Juglans nigra, Tribulus terrestris is a great GI antimicrobial botanical, even though most people in Western botanical medicine think of it for other things, like increasing endogenous testosterone production, which is really crappy at doing, but it's really good GI antimicrobial. Oil of oregano, often combining these types of things, the standardized botanicals and the volatile oil together hits the sensitivity spectrum that you want to kind of broadly treat dysbiosis. And they're not totally, but they're relatively sparing compared to rifaximin or some of these other agents at taking down all the good guys. So they spare more of the good guys at the dosage and frequency we generally use these natural agents. In other words, you have to use higher dosages and more frequent of the natural agents that I just showed you to take down a lot of the good guys than you do the bad guys. That's why they work so good. That's why those botanicals are inherently kind of selective for us. At the dosages we're using them generally, they're taking out the bad guys and they're mainly sparing the good guys, not entirely. I did want to mention thyroid here real quick though, because thyroid is extremely related to depression, right? If you're low thyroid, one of the symptoms, in addition to fatigue and coldness and constipation, low bowel motility and hair loss and all those things, the low bowel motility alone will shift your microbiota, right? But overtly depression, right? So often when we look at the thyroid, you'll see low thyroid function. In this person, they have overtly low T4 and they have functionally low T3. The free T3 is 2.4, right? A lot of times you see the T4s barely within the normal and then the T3 is either low or functionally very, very low on the low end of the range. And in this case, they have high thyroid antibodies as well. We know that thyroid autoimmunity is often triggered through the gut, through molecular mimicry with organisms that express peptides or protein structures that look a lot like thyroid tissue or TSH receptors in the case of Yersinia. So here's some papers on Yersinia showing saturable binding sites for TSH receptors. Basically, if you develop Yersinia overgrowth, your immune system is cueing in on some of the molecular patterns on the Yersinia surface, and they look almost exactly, not exactly, almost exactly like TSH receptor structure. So in a frenetic, over-amplified kind of immune system, you start taking out your own thyroid. And it's caused by a gut infection. Not always, because EBV, CMV, systemic viruses, and other things, we'll look in a second at another trigger for thyroid autoimmunity. But look at this sample here. Yersinia enterocolitica was overexpressed in the stool, but so was anti-gliadin secretory IgA. Gluten and gliadin antibody-antigen complexes have an affinity for the thyroid tissue and can direct an autoimmune response against the thyroid. And in the case of overt gluten intolerance, like frank celiac patients, they have 10 times the rate of autoimmune thyroiditis as non-celiacs. So it kind of demonstrates that connection. And people even without celiac disease, but just have what we would call non-celiac gluten sensitivity, maybe they were shown to have a high anti-gliadin secretory IgA, or what have you, they go off gluten for whatever reason. The beneficial effects that they experience often are not directly related to gluten or gliadin and an antigen-antibody immunological reaction. It's that when they go on a gluten-free diet, this study is suggesting, it fundamentally changes the composition of the dietary fibers in their diet and it shifts their microbiome to a more healthy place. So it doesn't matter, right? It doesn't matter what the mechanism is. If they get better, but that's interesting. And then finally, looking at barrier function as well. You see here another anti-gliadin secretory IgA, mildly elevated calprotectin, and mildly elevated fecal zonulin. Probably have a slightly inflamed gut lining, hyperpermeable gut lining. Maybe a big reason for this is they're eating gluten and they're immunologically intolerant to it, right? So some of these types of testing can, find these different ways and these different levels for you to kind of improve things for them. And if they have some barrier function issues, which they often do, you can use things like L-glutamine, which we know. But Dr. D'Adamo in his talk, if you saw it, was talking about glycomics. And this is a old-fashioned application of glycomics that comes really from naturopathic medicine principles, using things like mucilaginous herbs, slippery, slimy stuff, right? Like aloe, slippery elm, marshmallow, chamomile, okra. Did you ever make okra? All that snotty stuff in it, right? Aloe is slippery and slimy. Slippery elm, they even call it slippery, right? Those are the mucopolysaccharides in it. And the old time, botanical people would say, it coats your intestines and lets it heal. That's a good story. I'm not sure that's actually what's going on. I think these mucopolysaccharides are blocking antigen-antibody docking and knocking out immune amplification, right? Things like N-acetyl glucosamine, which Peter mentioned, I think, several times yesterday, and things like zinc carnosine. These are all things that can be very, very helpful. You can also use things like serum-derived bovine IgG, and again, N-acetyl D-glucosamine. The N-acetyl D-glucosamine is really cool. This is glycomics in action. And I'll show you this slide. Basically, when you're taking something like N-acetyl D-glucosamine exogenously, like in a powder, you're putting more of it in the gut. And what this does is it gets in, it kind of forms this lattice kind of network or shield around the binding sites. So you can't dock an antigen. The antigen-presenting cell can't dock an antigen in the T cell and activate it and cause immune amplification. It's like putting chewing gum in the padlock. You can't get the key in anymore, all right? That's what this stuff does, right? Is that right, Peter? Is that a good way to say it? Exercise can shift the microbiome. We know that exercise can increase bifidobacteria, lactobacillus. I wrote a whole paper on this in the Townsend letter, which you can either grab there or on my website here if you want to know more about that. Microbiome shifts associated with inflammation also can disrupt circadian rhythm, which means people don't sleep well. If you can't sleep and you don't get restorative sleep, it's hard to improve your mood. So you can use things like melatonin, 5-HTP, theanine. These can all be helpful in addition while you're fixing the gut. Keep in mind that psychotropic drugs, including antidepressants, shift the microbiome. And in fact, that's, like I said, that might be how they work to a large degree. They're antimicrobial generally, but some of them are anti-inflammatory. Anybody remember fluvoxamine being used in COVID? It's an SSRI, but it also affects the sigma-1 receptor and controls inflammation, reduces inflammation. That's why they used it in COVID-19, and they use it in long haul to some degree. And the SSRIs are not all strongly anti-inflammatory, so it's not a class effect. It's specific to fluvoxamine. But I just put it in there as an example of sometimes these drugs have more mechanisms of action than the one that they were approved based on and that we learned them based on. And also the gut microbiome also in alters the metabolism of these psych meds, basically. So when you shift the microbiome, you may shift the person's response to the prescriptive agent. You may even shift them from a non-responder to a responder, or from a responder to a non-responder. So you got to keep an eye on that. And there is studies, by the way, this is not a this or that. There are studies specifically, I put one here, on probiotic add-on therapy to SSRIs and other antidepressants. They work hand in hand with one another, and you get an added effect toward the beneficial side. So shifting from microbiome to metabolomics here a little bit, there's a lot of interesting elements on organic acid panels or larger panels, such would be considered metabolomics panels or small molecule panels. We're going to look at a few of these things here. The most obvious one is 5-hydroxyindolic acetic acid, or 5-HIAA for short. You can see that this is the downstream byproduct of detoxification of serotonin. You got L-tryptophan, not shown in the chemical diagram. Next is 5-hydroxytryptophan, and then 5-HT, which is serotonin. And then you go through monoamine oxidase, you get 5-HIAA. So when you're looking in the urine at 5-HIAA, it's a surrogate marker for how much serotonin was there. Okay, so very low 5-HIAA usually means you don't have enough substrate. You don't have enough serotonin to begin with. You need to prime the pump. How do you do it? With 5-HTP, right? Because if you use a selective serotonin reuptake inhibitor in someone who's not even making enough baseline serotonin, they're a non-responder. Because you can't block the reuptake of the serotonin that's not even there, right? There's some there, of course, or they wouldn't be functioning, but it's just the mechanism of the drug fails. That's why often you give them a couple hundred milligrams of 5-HTP and the drug starts working, right? Conversely, when 5-HIAA is high, I think it was shown there, that can also mean either you're really ramping up and relying on that serotonergic pathway, or often you're dumping it very, very quickly. And if you're a rapid metabolizer of it, it often means that you're left functionally with a lower level of serotonin. So in either extreme, it can be sometimes beneficial at least to do a trial of something like 5-HTP. And if you're using 5-HTP, you usually want a little pyridoxal 5-phosphate in there because that helps convert 5-HTP. The enzyme that converts 5-HTP to 5-HT, which is serotonin, is pyridoxal 5-phosphate dependent. Here's actually the chemistry. You see tryptophan, 5-hydroxytryptophan, 5-HT or serotonin, MAOI pathway, 5-HIAA excretion. That's where you're getting the metabolite right there at the end. We use 5-HTP because it's locked into this pathway. It can't go any other way. One way, serotonin, right? Tryptophan could go that way, but it can also go other ways, right? And in people who are in inflammatory stance, they're immune activated, they tend to want to favor going this way, which is the kynurenate pathway. And that's not an inherently good thing. And we'll look at that. And by the way, microbes in the gut can alter this biochemistry and push it toward the kynurenate inflammatory and neuroexcitatory pathway versus going to serotonin. So what we want to do is we want the tryptophan to go toward 5-HTP and serotonin, shown in the green little oval. We don't want it to dominantly go down to kynurenate and quinolinate. These are molecules that are found at high levels in inflammatory conditions, and you see, for instance, very high kynurenate, quinolinate in end stage of life. You see it after traumatic head injury, you see it in septic shock, you see it in all kinds of different things. It's used as a marker in critical care medicine for that reason, okay, kynurenate and quinolinate. But it's a double-edged sword, as most things are. Quinolinic acid, or quinolinate, is also an essential precursor for NAD+ biosynthesis. So it's not all bad, or it probably wouldn't be there, right? So it's never that easy of a story, but it's a matter of how much, when, and a balance. So you don't want to totally shut off the kynurenate quinolinate pathway, but you don't want it overfeeding that way either, or it can create central nervous system excitation and inflammation, which can lead certainly to anxiety, insomnia, panic attack, and depression. Here's some studies on actual depression right on point and increases in quinolinate and activation of that pathway. And you can see here on a metabolomics panel, look at here in the first box I highlighted, tryptophan is low, 5-HIAA is low. If you look at that in isolation, you're going, wow, they don't have enough serotonin. They have no 5-HIAA. What's a precursor to serotonin? Tryptophan. Holy God, tryptophan's low. What do I need to give him? Tryptophan, right? Not really. Where's tryptophan going? Look at the bottom. Kynurenic acid or kynurenate and quinolinic acid are high. The tryptophan is not going this way to serotonin, it's going that way into the inflammatory metabolites, and it's throwing off the hydroxykynurenine and ultimately what's called the KT ratio, the kynurenine-tryptophan ratio. This is an excitatory state metabolically in the central nervous system. This is not a person you want to give tryptophan to. This is a person you want to calm this down. The most direct intervention besides stress management and all the biofeedback and all those different things, the most direct thing from a administration of a biochemical model in natural medicine is fish oils, omega-3 fatty acids. Okay, you can also use phosphatidylserine, phosphatidylcholine, things like that. Okay, but you also want to fuel that serotonergic pathway to calm things down, and you do that with 5-HTP. Keep in mind, quinolinic and kynuric acid can be temporarily upregulated, and you'll find it high in a lab test if someone has a viral infection. So keep that in mind. That might be a temporary, just normal physiological response, and that includes COVID-19. These are just some kind of facts on quinolinic acid that I kind of talked about earlier. Then we have two other really cool metabolites to look at, homovinylic acid and vanilmandeleic acid, which is HVA we call, and VMA. These are catecholamine downstream products. So HVA is the downstream product of dopamine. Vanilmandelic acid, or VMA, is epinephrine and norepinephrine. Okay, so if they're really low, like shown here, this is often associated with adrenal fatigue, where someone is just, they're flat out fatigued. They got nothing left. They can't get off the couch. They generally don't have anxiety, panic attack, and any of that stuff. They don't have enough energy to have anxiety, right? They're just lights are off, right? These are the key people that you got to really build their adrenals back up. You got to give them, even for a while, low dose like Cortef or something like that, just to bring the lights back on, okay? There's a definitely opposite pattern here, which is high VMA, high HVA. This is where the catecholamines are being overproduced, and you're seeing the downstream products of it. This is someone who may have fatigue, but they're wired and tired, right? This is more common, at least in my patient population that I see, because I see a lot of people with global pain syndromes, fibromyalgia, chronic fatigue, ME, long haul COVID. They generally have this pattern, high catecholamines, and this, you see this a lot here. High catecholamines, HVA and VMA, low 5-HIAA, elevated cortisol. This is classic PTSD metabolomics pattern, okay? But you don't only see it in frank PTSD. You see it in classic fibro. You see it in IBS. You see it in long haul. You see it in a lot of these things. Because, a lot of these chronic disorders that we see and that are very difficult, there's no real definitive cure for. Many of them are post-viral syndromes, as we know. Long haul COVID is just the latest manifestation of it, but chronic fatigue, ME, classic true global pain syndrome like fibromyalgia, mainly post-viral syndromes, right? And it's locking the immune system in this aberrant pattern of hyperactivation, microglial inflammation, brain processing gets all messed up, stress response gets all messed up, and they get into this PTSD pattern. So you see this pattern in a lot of different disorders, because those disorders are just different trapdoors where people fall into the same pit. And that's potentially a good thing, right? Because I've been practicing 30 years. For 30 years, I've seen patients with chronic fatigue, ME, fibromyalgia, all this stuff, just basically ignored by the medical system and told they have a Prozac deficiency eventually, right? And then they come to providers like us, and we do all the things we can do to help them and make them better, even if not making them perfectly cured, if you will. But long haul COVID came along. It's affecting so many people in a very similar way. They can't ignore it anymore. And there's so much economic ramifications that are coming because of it. And there's so much money to be made because of the numbers. Now there's massive interest in it. And research is going on incredibly fast on it. I'm at the point with these disorders, I don't care what fixes it. I don't care if it's a vitamin, an herb, a drug. It could be cobra venom. I don't care. I'm agnostic to what it is. I just want it, right? Because these people are suffering. But I do think, since all this effort is being put into long-haul COVID, if they find an agent or an approach that works in long-haul COVID, I'm pretty confident it's going to work in chronic fatigue and ME and fibromyalgia and all these other things. And it may be really transformational for a lot of people. At least I'm hoping so. Okay, let's go forward. So this is a classic PTSD type of pattern right here. And you guys know how to calm this down. Things like, GABAergic botanicals, passion flower, lemon balm, valeriana, calming adaptogens like Withania somnifera or ashwagandha. Withania somnifera, right? Somnolence, calming. Other adaptogens are not. I'll show you those in a minute. Taurine, theanine, some B vitamins and magnesium is just sort of adrenal food in general for stress. Or another example here, German chamomile, L-theanine, 5-HTP, phosphatidylserine. These are all calming adrenal therapeutics for people with those high catecholamines, right? Wired and tired. On the other hand, when things are low, when things are just flatline, VMA is low, HVA is low, serotonin's not working, cortisol is not elevated. It's just lights aren't on. Then you need to build it up. Then you're looking at things like Eleuthero. You're looking at things like rhodiola, giving tyrosine. These are adrenal tonics, right? You don't want to give this type of adrenal product to the wired and tired elevated catecholamine person. You will make them worse. You will cause panic attacks and anxiety. And a lot of those people go into the health food store, and what do they give them? Oh, you sound like you're really stressed. Here's our stress formula. And it's full of B vitamins, tyrosine, and stimulating adaptogens, right? And they get worse. If you had metabolomics, you wouldn't do that. You can look at GABA as well. And of course, GABA, we can directly kind of help look at that glutamate versus GABA, excitatory versus calming, you know, how they ratio out right there. And if necessary, give GABA, right? And use a fermented GABA, and often in a chewable form that can act very, very quickly. And then finally, on metabolomics, we can get at least an idea of potential microbial dysbiosis and overgrowth by looking at all these different metabolomic or organic acid compounds that are produced not by us, but by gut organisms based on breaking down certain food substrates. And if you're producing lots of certain ones at certain levels, it's indicative of a microbial composition that's out of balance, that's got overgrowth of opportunistic organisms. And then finally, just touching on genomics here, this is, I'm going to show you some things from Dr. Diadamo's Opus 23, an iteration of it known as GenomicInsights by DSL. But the Opus 23 goes out and looks at the medical literature for you, and PubMed and all that, but it looks at all these different metadatabases to make conclusions and to compare things, to look at what all the science is saying about not one SNP at a time, but what the value here is looking at a multitude of SNPs in a network, right? How everything comes together in pattern analysis. So it's not just reductionist, like this SNP says this, so give this, right? Often one SNP alone with a polymorphism doesn't equate to much. It's just looking at a higher propensity for something to happen. It doesn't mean it's happening. With metabolomics, you're measuring what's happening. With genomics, generally you're measuring what's more likely to happen or less likely to happen. Doesn't necessarily, or some exceptions to this, doesn't necessarily mean it is happening, right? So it's very, very different. But if you combine them in this multi-omics look, it can be much more powerful. So I'm in GenomicInsight here, which is Opus 23 Explorer. I just in the search box put catecholamines. I'm interested in catecholamines, right? Stress hormones. Catecholamines, boom, all these different genes and then sub-SNPs come up. And the first gene there of the highest magnitude is COMT, right? So you go, well, I don't know that much about genomics. What is that? Well, you can just click on COMT if you don't know, and the program teaches you about COMT, okay? Catechol-O-methyltransferase gene helps break down neurotransmitters dopamine, norepinephrine. A defect due to certain variants in COMT will cause higher levels of dopamine due to slower breakdown, which can contribute to anxiety and insomnia. Individuals could be more susceptible to dopamine fluctuations and therefore mood swings, right? So if you see SNPs that are non-beneficial alterations in the COMT pathways, you might have to support that. Okay, if you want to look at another gene related to catecholamines, MAOA, monoamine oxidase A breaks down neurotransmitters. This is going to determine how rapidly you break down your neurotransmitters or not. So this can lead to low or high levels of these neurotransmitters, which can cause mood swings, OCD, anxiety, aggression, insomnia, depression, and you get the idea. And it says here the enzyme required for function here is B2, or riboflavin, so you need to make sure that's sufficient. So these are just ways you can hone in. And if you want to go more and look at it on a network, kind of metabolic network basis, you can do something like just select one carbon metabolism. We know methyl tetrahydrofolate and methylation is very important in depression because of its role in neurotransmitters. And instead of, like a lot of things, you'll just get MTHF, R and a couple of SNPs there, right? This shows it to you on a whole metabolic diagram, flow diagram. So you can look where the orange is the problems, right? And the higher the degree of orange in the box, the bigger the problem. So right here, you can just hone in. Well, there's something going on in methyl tetrahydrofolate reductase, but there's a couple of other genes there I might want to look at. But when it comes just to methyl tetrahydrofolate reductase, you know, the two big stars here, the C677T and the A1298C, are shown there. And you can see the risk allele, the type, it's a risk SNP, what the patient's outcome is, and are they homozygous, heterozygous, et cetera. You can see here, it's a bit dirty, right? This, the C677TT is plus, and the 1298C is minus plus, so it's kind of a half risk there. But when you figure those out together and you do a multi-SNP macro analysis on this, that pattern there, the plus on C677T and the plus minus on 1298C, ends up in a 60 to 70% reduced enzyme activity right there. That's significant, right? This is someone you want to make sure you're supplementing along this pathway. So methyl tetrahydrofolate, either methyl or adenosine cobalamin, methyl groups, potentially B6, things like that. And you can put this out on a report to talk to the patient about it on the client side report. You could also dive in and have the program tell you what are the best agents to use. The ones shown here in green will affect one gene on this diagram. The ones in blue are affecting multiple, or two or more. So you can, if you had problems on methyl tetrahydrofolate reductase, like our example did, but you also had problems up on MTRR, 5-methyl tetrahydrofolate homocysteine methyltransferase reductase, you can see now why they call it MTRR, you can pick some of those things in the blue, because it's going to kill two birds in one stone. And you see these are things like we would probably gravitate toward using 5-methyl tetrahydrofolate, cobalamin, betaine, B6. But you can even have the program do more of your work for you and just hit the button and it tells you which things to use based on the PubMed literature searches and other medical databases. And they're ranked. The program has no biases, right? The program is just looking at the weight of the evidence in the medical literature. So what comes out number one in this methyl tetrahydrofolate reductase SNP issue? Vitamin B9 or folic acid. Look farther down. I think it's number 9. 5-methyl tetrahydrofolate. Well, you go, well, that isn't right. Diadamo's lost his mind. Everyone knows you use 5-methyl tetrahydrofolate, not synthetic folic acid, right? But the program doesn't care about that. The program just looks at how many scientific papers have supported the use of what. And folic acid's been around a lot longer than the common use of 5-methyl tetrahydrofolate, and therefore it wins the battle, right? Because it wins in the literature. So that's where you need to also know, hey, the better form of folate there is 5-methyl tetrahydrofolate, okay? We can look at some other things on metabolomics, like if we're interested in B12, instead of serum B12, of course, we can just look at the methylmalonic acid, because we know when you convert methylmalonate to succinate in energy metabolism, if the enzyme that's responsible for that conversion, I think it's succinate beta synthase, I think, is B12 dependent. So if you're functionally B12 insufficient, you get a buildup of methylmalonate and it spills into the urine. Or you can pick it up in the blood. It's also just a test available at Quest, serum MMA. The analogous marker and here's an elevated MMA, the analogous marker in folic acid land is Figlu. But by the way, B12, there are studies showing that MMA, either in blood or in urine, is much more sensitive and able to pick up subtle B12 insufficiencies than any of these other things that you would think of, like low serum B12, hypersegmentation of RBCs, macrocytosis, megaloblastic marrow, and then actually overt anemia. So you're picking it up way more functionally with an MMA. Here's Figlu for folate, and these appear. Here's Figlu being high on a organic acid or a metabolomics. This is an omics panel from DSL, and you can see here this would be a direct objective evidence to give them folate. And you can use MMA, folate, and homocysteine, 3 different tests, and it can actually help you exactly triangulate on which nutrients the person needs. Do they need just B12? Do they need just folate? Do they need B12 and folate? Do they need just B6? Do they need folate and B6? And there's a little matrix there for you, and I don't have time to go over it, but by using all three of those tests, you can zone in on exactly what they need. Or you can just sort of give them a formulary thing that has, 5-methyl tetrahydrofolate, some methylcobalamin. This is actually a natural product for mood that has some good clinical outcome studies on it. It has the B12 folate, but it has 2 botanical extracts, saffron or crocus and scaledium. And these are specific proprietary versions of these botanicals that are standardized, and they've been used in clinical trials. That's why I'm showing them here. The Zembrin, which is the scaledium, has really good studies on it, showing a dual mechanism of action in depression, acting as both an SSRI and a PDE4 inhibitor. PDE4 inhibitors inhibit microinflammation, deep tissue brain inflammation. And the saffron extract here, which is Crocus sativus, also has some really good clinical outcome studies on mood. This is one of them. This is Crocus direct head-to-head comparison with citalopram. And it basically showed equivalent results. Pretty amazing. So what I was just showing you here is a little taste or a smattering of using multi-omics. We looked at some SNPs, and I only showed you a couple of SNPs. There are more involved in mood and depression and anxiety. But things like some SNPs or genomics, some microbiomics, some metabolomics, all of these can be done with non-invasive samples. So buccal swab for genomics, stool for microbiome, and you can do most of the metabolomics through just urine. And it can really help you target your therapies and kind of keeps you in the future lane, because this is where things are going. It's no more this for that nutrition. I'm giving this for that because it's a protocol. Protocols are limited. They're generic. They're sort of vanilla approaches that are not personalized. And pretty soon, they don't need you to do that, because that can all be done on the internet through some direct-to-consumer precision medical play. They need you to do this kind of stuff, because you can't automate all this, at least yet. Okay? So doing this kind of thing in your practice is really kind of where everything is going to go. So thank you. I hope it was worth your time. I'm going to stick around for some questions in the back and enjoy the rest of the conference. Thanks.